Hi everyone. We'll change gears in the course, and last few lectures we have devoted to deriving an expression for speed of evolution in analytical form, and making some assumptions. We were able to come up with an order of we were able to come up with expressions that would give us order of magnitude estimates for speed of evolution in a simple microbial experiment that we might be doing. We will talk about a different aspect of evolution associated with evolutionary analysis, and this is called evolutionary game theory. And the simplest way to understand this is that so far we have been talking about fitness of a particular genotype in the sense that the fitness of a genotype is independent of what are the biological entities that are surrounding it. But very often what happens is that one particular genotype is interacting with another genotype present in the environment, and hence the fitness associated with this particular genotype is closely linked to what all other genotypes are present in the environment. In that sense, fitness of genotype A is not just dependent on A itself, or is not just a constant associated. As completely opposite to this, fitness of genotype A is actually dependent on the frequencies of genotypes A, B, C, and however many genotypes that are present in the environment, and also what are the relative frequencies associated with these genotypes in the environment. So, fitness becomes a complex thing to analyze in a setting like this, and that is what we will spend the next few lectures in this course on, and trying to come up with systems as to try to come up with analysis and understand how do we look at these systems and understand them. So, let us start this thing, and this particular section is going to be called evolutionary game theory. And the premise behind this is that the fitness of a genotype, let us say A, is not constant but depends on the relative frequencies of the different phenotypes that are present in the environment. So, that is evolutionary game theory, and now we are going to have these genotypes which are going to be constantly interacting with each other, and their fitness will be dependent on the sum total of what is the current composition of the population at any particular instant. But what that also means is that a particular genotype is going to want to try and acquire mutations, any mutation that increases its fitness. So, if I am genotype A, I am going to try any mutation in my genome which makes me take advantage of other genotypes in a better way, will lead to increase in my own fitness, and hence I will change my genotype so that I can exploit other genotypes better. Similarly, other genotypes, any mutation in them which make use of what I am offering to the environment better is going to be favorable and selected for, and hence those genotypes are going to change and evolve strategies as well. So, you have these constant evolution of strategies between these two particular genotypes, and that is the sort of a dynamics that we are interested in analyzing under evolutionary game theory. Okay. So, let's start with an example associated where something like this might play out. Let us imagine an environment. Let us imagine an environment where food is scarce. So, this is my environment E, and in this environment food is scarce, and the only food that is sort of available is these polymers in the environment which cannot be directly used by bacteria. So, these are polymers which are not directly used by bacteria, and what is going to happen is that, and these are the bacteria which are present in the environment, and these extracellular polymers cannot be used up by the bacterium. But what is going to happen is that the bacteria in the environment are going to produce an enzyme, and these enzymes are produced in the cytoplasm of the bacteria and released outside the cell, and the job of these enzymes is to break down these polymers into little monomers. So, the polymer gets broken down because of this extracellular enzyme, and that broken monomer is then taken up by the bacteria and used for growth. So, this is the scenario that we have, and all particular individuals in the population belong to this particular genotype. Now, what might happen is, well, one way to look at this is that every single individual in this population paying a certain cost in terms of the energy investment that it is making towards production of this enzyme, which is not directly benefiting the producer. The enzyme which is produced is not breaking down the polymer outside just for my sake, but the enzyme that I am secreting is going to go into the environment and break the polymer, and that the broken monomers can be used by any individual belonging to the population. So, in that sense, the enzyme that I am making by using my own private goods and I am releasing into the environment is doing a public benefit in the sense that it breaks down the polymer and creates monomers which can be used by any individual in the population. So, this is a private investment that I am making, but the benefits associated with this private investment are public in nature, in the sense that any individual belonging to the species can use the monomers which are broken down by this enzyme. So, that is the setting that we have, and of course, every particular enzyme molecule which is released into the environment is doing the same, and bacteria are taking up these monomers and using them for growth purposes. But what is going to happen is that, let us imagine that one of the bacteria living in the environment acquires a mutation. So, this is my mutant in the population now, and what this mutant is doing is that it has picked up a genetic mutation which no longer leaves it capable of producing that enzyme. So, this particular mutant is not producing the enzyme, but it still retains the capability of picking up the monomers from the environment and using them for energy. Now, imagine this one mutant bacterium in a pool of normal bacteria. Every single individual is paying a certain metabolic cost towards production and release of the enzyme into the media. However, the mutant that we are talking about here is paying no cost towards production of the enzyme, but is only reaping the benefits of the enzyme that has been produced by others. So, in a setting such as this, because my mutant is not paying any cost in terms of production of enzyme, and every other individual in the population is paying that metabolic cost towards production of this enzyme, fitness of the mutant is more than fitness of what we will call the parent genotype. Now, we have seen what happens when something like this happens. Now, we have two distinct genotypes in the environment, the mutant genotype and the parent genotype, and the mutant has a fitness more than the parent because it is not paying any cost towards production of a public good, the enzyme which helps provide food for every individual in the population. Because there is a fitness differential, selection will take over and the frequency of the mutant is going to increase in the environment. And now, because the mutant is fitter than the wild type, the way selection will act is the numbers associated with the mutant are going to increase and wild type are going to get, the parent genotype is going to get sort of eliminated from the environment. And if that happens, let us take a look at the picture that will be offered when that is the case. After selection has acted for a while, the environment is going to look something like this. So, this is the same environment E. This environment has food resources. These are the polymers that can't be broken down by the mutants or the wild type, but can be broken down by the enzymes that are produced by the wild type. And these are the bacteria now. And because selection has acted for a while, most of these bacteria belong to the mutant genotype. Let us imagine the scenario in this case to be something like this, where most genotypes are mutants and only this one parent genotype survives. Now, this parent genotype is still producing the enzyme, but of course, because there is only one producer of the enzyme production will not be very much, and as a result the polymer that is broken down only be in the vicinity of this of the location where this parent genotype individual still exists. So, now in this case what is going to happen is that because this a setting like this has been arrived at, most of the benefit that is obtained from breaking down this polymer is going to be derived by this parent genotype only. These mutants are not going to see the monomer at all and are going to get selected against, whereas these mutants may see some amount of monomer associated with the polymer because of their physical proximity with the parent genotype. But as a result of all of this, what this means is that in this case now, the fitness of the mutant is now less than fitness of the parent. Okay. And what this sort of illustrates is that if we were to start with an environment that we started with, there was only one mutant and everybody else belonged to the parent genotype was producing the enzyme. In that case, fitness of mutant was more than fitness of parent, and eventually this environment transitions itself to the second environment that we drew. Selection acts on the two genotypes and we have mutant population which now dominates over the parent genotype, and in this case fitness of mutant is less than fitness of parent. So, what all of this illustrates is that change in frequency, the frequency of the mutants in our example went up and the frequency of the parent genotype went down, and this change in frequency, what it resulted in was that the fitness of mutant, which was more than fitness of the parent to begin with, now became less than parent, and this change was facilitated just by changing the genetic makeover or the genetic or just by the frequencies associated with the various genotypes that are present in the environment. So, again fitness in this sense is a concept associated with the frequencies associated with the different genotypes in the environment, and they are not fixed in nature. Okay. So, how do we look at that? Remember in the first few lectures, of course, when we started dynamics, capturing dynamics in a mathematical form, we would write. So, again let us go back to our simplest example that we started with, two particular genotypes, and we will call them A and B. So, if we have two genotypes, let us call them A and B. Let the frequency of individuals belonging to genotype A be XA, frequency of individuals belonging to genotype B be XB, then we know that XA plus XB is equal to 1. And what that means is that the composition of the population of the environment is denoted by this vector X, which is XA and XB. If there were more genotypes, this vector would just have more components, but for our example, this vector denotes the composition of the population at any given instant in time. Also, because fitness is now a concept which is dependent on frequencies and not a constant, fitness of A is given by fitness of A, but this is a variable which is dependent on composition of the population, and composition is defined by the vector x hat. Similarly, fitness of B will be a variable quantity which is dependent on the vector x. So, this is fitness of genotype A. and fitness of genotype B. Okay. All right. So, if we have this, then we can write that x A dot is just equal to x A into fitness of genotype A, which is f A function of vector x minus phi, and x B is just equal to x B dot, which is just, this is just x A dot is just short for dx A by dt, the rate of change of frequency of number of individuals belonging to genotype A. X B dot is just dx B by dt is equal to x B f B x minus phi, and phi of course is the mean fitness associated with this environment, which is just going to be equal to x A times f A x plus x B times f B x. So, we can simplify this further. We can plug this value of phi in the first expression that we have here and get dx A by dt is equal to x A into f A into x minus x A into f A x minus, and I am going to write x B as 1 minus x A times f B into x. That is the expression that we get, and we can simplify this further and write this as dx A by dt equals x A into f A x into 1 minus x A minus 1 minus x A times f B x, which can be simplified as x A times 1 minus x A times f A x minus f B x. That is the dynamical equation which defines the dynamics of these two genotypes, where fitness is not a constant, but a function of the composition of the population. So, if we have this, now the next thing associated with this is just going to be the analysis of the steady states and the stabilities associated with this system. So, again we'll use the standard strategy that we've been doing so far. What are the steady states of this system? And steady state, by just looking at this, we can tell is equal to x A equal to 0, which means that it's only B in the environment, or x A equals 1, which implies that there is only A in the environment, or any such scenario or any such composition such that f A of x is equal to f B of x. What we mean by that is these are the two compositions of the environment where dx A by dt is equal to 0, but these are intuitive in the sense that this particular value x A equal to 0 is talking about the case where there is only B in the environment, and x A equal to 1 talks about the case where there is only A in the environment. So, these are steady states, but in addition we also have another steady state which is given by that composition x such that f A of x is equal to f B of x, which means those compositions where fitness of A is exactly matched by fitness of genotype B. And now, so we may need to solve for these fitnesses depending on what are the exact expressions of these. Now, let's think about this a little more. Imagine a case where f A x minus fbx looks something like this. So, I am going to plot, this is x, and on the y axis I have fa of x minus fb of x, and let us say this is xa, this is 0 and this is 1. So, again let us imagine, so this will be all a's, this will be all b's, and this is fa and xb at every particular fax minus fbx at all particular values of the variable xa. So, if this quantity looks something like this, so imagine that as xa varies from 0, which represents the scenario that there are only b's in the environment, and goes towards xa equals 1, which represents the scenario that there are only a's in the environment. If the quantity fax minus fbx, which is the difference between fitness of genotypes A and B, that expression looks like this, which is not constant but is changing with composition. If that looks like this, what is the stable steady state of the system? Okay. So, in one sense we have already talked about this. So, maybe pause the video for a minute or so, think about the system and continue thereafter. But what you should realize here is that our equation that we are working with is the following, which is dxa by dt is equal to xa times 1 minus xa times fax minus fbx. And what this tells me is that xa is a quantity which is always greater than equal to 0. This is equal to 0 when xa is equal to 0, but otherwise is always greater than 0. So, it is a positive quantity. 1 minus xa is an expression which is always greater than equal to 0, because by definition xa cannot exceed 1. So, 1 minus xa is equal to 0 when xa is equal to 1, but is otherwise between 0 and 1 and is always positive. And fa minus fb is in this case given to me by this expression, which tells me that fax minus fbx is type of a function which is always greater than 0. So, what that tells me is that this expression dxa by dt is actually never less than 0, but could only be equal to 0 or greater than 0. It is only equal to 0 at two particular values, xa equal to 0, xa equal to 1. But at any other value of xa, between these two extremes, if my system was present at any of these two values, my dxa by dt will be a positive number times a positive numbers times another positive number. That means dxa by dt will be greater than 0 as long as xa is not equal to 0 and xa is not equal to 1. For any other value of xa, dxa by dt is greater than 0. And what that means is, if my system is at this place, then dxa by dt is greater than 0, which means as time increases xa increases, and hence my system moves in this direction, and eventually it stops at xa equal to 1, because that is the value where dxa by dt is equal to 0. So, this is the stable steady state associated with the system. And xa equal to 0 is the unstable steady state. So, this is analysis of stability using something that we have already done in the course before, but if you think about it, this makes a lot of intuitive sense. Let us take a look at the kind of a function that fax minus fbx is. if you look at this graph, then Fax minus Fbx is always greater than 0. What that means, because this graph Fax minus Fbx is always greater than 0, and that greater than 0 is independent of the composition of the environment. So, no matter what is the composition of the environment, Fax minus Fbx is always greater than 0. What that means is that if I were to start my environment with a composition anywhere on this scale, fitness of A minus fitness of B is greater than 0, and what that means is that fitness of A is more than fitness of B, and what that means is that selection is going to act and select for genotype A and remove genotype B, because Fa minus Fb is a positive numbers, which implies Fa is more than Fb. So, A is growing at a rate which is greater than the rate at which genotype B is growing, and that fact is independent of the composition of the population in the setting that we are talking about right here. And if A is fitter than B irrespective of the composition, it is only natural to expect that selection would totally eliminate B from the population, and eventually you will have only A's survive in the population, which is what our stability analysis is telling us right here. Okay. So, let us quickly take a look at the converse of this situation as well. If Fa minus Fb was a graph which looked like this is Xa going from 0 to 1, and now if my graph looked something like this, what that tells me is that Fax minus Fbx is always less than 0, and this fact is independent of the composition of the environment, which tells me that Fax is less than Fbx independent of the composition, and if B is fitter than A, then selection should automatically act and ensure that A is driven out of the population. So, if you do the stability analysis, I leave that as an exercise for you. If you do that analysis, the population should move towards left and approach 0. And what that tells me in terms of my steady states associated with the system is that Xa equal to 1 is an unstable steady state. And Xa equal to 0 is my stable steady state. So, when we started this discussion, we said that there are 3 types of steady states that might occur in the system, and those 3 types of steady states are Xa equal to 0, Xa equal to 1, and also all those points where any such composition which ensures that f of A is equal to f of B will also be a steady state according to this situation, because if this is satisfied for any composition of x, then dxa by dt will equal to 0, because this quantity then becomes equal to 0. But so far the 2 examples that we have done, which is this one where Fax minus Fbx is always greater than 0, and the other one where Fax minus Fbx is always less than 0, do not offer us any such example where any such composition where Fax is equal to Fbx for a particular composition associated with the environment. And that is something that we will start with in the next lecture. Thank you.